Hello everyone, my name is Savely Komlev or Sava, and today I would like to present to you my project on AI-driven pipeline for intrabody design, which focused on predicting and optimizing intracellular stability of nanobodies. It was done as a master thesis for Paris University in collaboration with Argentis, which is a small biotech company in the United States. Let's start with a bit of background. So antibodies, I'm sure everyone heard about them during the pandemic. They are a specific kind of proteins which are produced by an organism, for example, by a human, to in response to some toxins or pathogens like viruses or bacteria entering the body. And the goal of antibodies is to bind with a specific part of their structure called antigen binding fragment to antigens, which are parts of the pathogens they target. So their goal is to bind to, say, set pathogens. For example, like on this picture, four antibodies bind to a COVID-19 virus and they deactivate it. For the sake of this project, we can think that there are two ways, main ways to present antibody computationally. First, we can present it as a set of atoms with their coordinates relative to each other, and we get a 3D structure of this antibody, or we can present it in a much simpler way as a sequence of amino acids that make up this antibody, and in this way it will just be a short text. There is a specific kind of antibodies, which is intrabodies, and intrabodies are not, they don't occur naturally, but they can be designed artificially to act within cells. As you can see on this schematic picture, conventional antibodies, they act outside of cells. Here it binds to a virus, for example. But intrabodies are designed to act within cells, which allows them to access targets not accessible to common antibodies. For example, here it binds to a cancer protein. And it's beneficial because it allows us to target some harmful things inside cells, like cancer proteins or proteins associated with neurodegenerative diseases like Alzheimer's or Parkinson's disease, or even viral infections like HIV. The main problem with intrabodies is that they need to be able to remain stable in a harsh reducing environment of the cell because the environment for conventional antibodies is different. So when you design intrabodies, like one of the main bottlenecks is to make them stable inside this specific environment. Let's move on to the problem and solution. So the problem is that current intrabody design relies on using some ad hoc computational tools and wet lab screening of thousands of candidates. And this process is very slow. It takes months to screen those candidates, and it's expensive. You spend tens of thousands of dollars per one round, and you usually need several rounds of generating and selecting intrabodies. And our solution is to use modern AI tools to build a pipeline which helps to generate a lot of intrabodies and select only the best of them. And then we can test only those best candidates in a wet lab. Thus, we will cut down the costs and time by an order of magnitude. And here is the whole pipeline. As you can see, as an input, it takes an antigen, like a target, and a few intrabody candidates, initial candidates, which don't have to be good, but we need to start somewhere. Then we use different methods to generate a lot of uh, different interbody candidates. Then we select them by sequence properties, and then we select them by structure and interaction properties. And in the end, we get a few optimized interbodies that we can then test in the lab. So here, my pipeline focuses on the blue parts, on making stabilizing mutations, and on predicting stability of interbodies. Now let's move on to the stability prediction part. So the approach that I ended up using is displayed on this figure 6. We take intrabodies as an input. We represent them as sequences. We then remove highly variable regions from them. Uh, it's the rationale here is biological because in the antibody you have different regions, and some of those regions are highly variable, and they are mostly about binding to a target, while some regions are more conservative and consistent across different sequences, and they form a sort of backbone of this antibody, and they are more relevant to stability. So using this information, we removed highly variable regions because according to our research, they introduce more noise and signal in this task, and we ended up with those cut sequences, which we then input to transformer protein language models, which the architecture is similar to the large language models, but it was trained on billions of proteins and learned sort of evolutionary rules of how proteins are made. And then we can use embeddings from those models to build classification machine learning models, which help us to classify intrabodies to either stable or unstable. It's a supervised learning problem, and to construct a data set, I went through tens of research articles, and the main constraint here that we faced is that the scarcity of experimental data. I was able to find less than 200 labeled sequences for this task. Let's move on to feature engineering and architecture choices. So first, I compared fine-tuning with LoRa versus just building a classifier on top of a frozen embeddings of those large protein language models. Here you can see the results of comparison. And in terms of precision, F1 score and ROCAUC, you can see that frozen embeddings perform better. The only benefit of using LoRa was in recall, but in this task we care about precision far more, and we want a more balanced model in the end. Plus, the frozen embeddings is computationally less intensive, so we choose to move with frozen embeddings. And I believe the main issue with LoRa here is the dataset size, so it just overfits easily. Here, I compared using full sequences versus removing highly variable regions, because those highly variable regions likely introduce noise to this task rather than signal. And here, those cut sequences are represented in blue and full sequences in orange. And you can see that pretty much regardless of the architecture, we have better precision and recall on the cut sequences. Here, I compared different kinds of protein language models. ESM models are developed by Meta or Facebook. Proti5 was developed by a research group from Germany, and IGT5 is just a fine-tuned version of Proti5 specifically for antibodies. Here you can see that as we go from the smallest model in blue to the largest model in yellow, is like the green and brown being middle-sized. Generally, the dependency is that the more parameters we have in the model, the better performance is. But it stops working when we move on to the model that was specifically fine-tuned for antibodies, the IGT5 model. It's smaller in terms of parameter, more than 2 times smaller, but it performs just as well as the biggest 3 billion parameter model here. So fine-tuning is important, and we moved on with this fine-tuned model. I also integrated structural information into the model. So I just fused the frozen sequence embeddings with structural confidence scores uh, predicted by another tool, ESMFold. So basically, I used an external tool to predict confidence of each amino acid position in the antibody 3D structure, and as you can see here, it improved recall by 8%. So we were able to compensate for the recall lost we had when we choose to not use LoRa. And those in the end, our best model, random forest IGT5 on frozen embeddings plus structural scores, we had a rock oak of 81%, which is quite a good result for this task. I also tried to add some physicochemical properties like length of the antibody, its charge, hydrophobicity, average flexibility, molecular weight of this antibody, but it didn't improve the performance, so I suppose that those properties are already encoded in the language models embeddings. And turns out that yes, I was able to identify specific embedding dimensions which highly correlate to those properties. So they pretty much encoded there, and it offers some degree of interpretability to the model. Now let's move on to not predicting the stability, but to optimizing it. And here I performed a statistical analysis which revealed that some specific positions in an antibody sequence, like for example this position 54, where if we have a letter A, amino acid A at that position, then it's much more likely to be a stable antibody, intrabody. While if we have a letter S at this exact position, then it's much more likely to be unstable. So just like gathering this statistical information, I made a probabilistic consensus-based mutagenesis algorithm, which basically takes like a parent intrabody. It extracts only those framework parts of it, non-highly variable parts of it, and then it assigns mutation sites based on this statistical information. So it's more likely to select for mutation the sites that are likely to lead to instability. Then it decides which amino acids to mutate at this position. So like it's more likely to mutate the unstable to something else, or like unstable to a stable amino acid at this position. If we have data for that in the database that I constructed, and in the end, it just generates a bunch of variants with different mutations where if we have data, those mutations, for some mutations, those mutations are just more likely. And here you can see the results. In blue, the initial candidates that we have for survivin, which is a cancer-related protein, and in yellow are the same results measured for the optimized candidates. And as you can see, for example, here on AB intrabody score, which intrabody stability, here we can see that in initial candidates, we had a 30% probability of them being stable as the highest one. And after making mutations, we get a cohort of intrabodies with up to like 85% of stability probability. Here I measured PODDT, which is a structure confidence score, and you can see that the mean for optimized also shifts towards better scores. So this is like another independent metric that I used to confirm that this algorithm works. And the third metric that I measured is a disorder, which was measured by another external tools. And here you can see that again, statistically significant change in optimized mutants towards less disorder, meaning more stability. And that's what we want. So let's move on to limitations, perspectives, and conclusions. Here is the UMAP in protein language model embedding space. And as you can see, there are no like defined clusters of stable and unstable antibodies in this space, which suggests that this task is like quite complex and probably we can't resolve it with 100% accuracy. But I think that the main problem is that we had very limited data set, less than 200 sequences. We were not able to train more complex machine learning models and had to resort to frozen embeddings. So I think that with more data and using more complex models, we would be able to push this boundary. But for now, it's like 80% accuracy. And perspectives, we used our pipeline to generate more than 5,000 candidates that are now being tested in the University of Maryland. And some of those were like very promising. And we hope that after the experiment ends and we will receive like more than 5,000 sequences, we will be able to train more complex models. Thanks for attention and feel free to connect with me on LinkedIn.